Welcome to Biology 2402 Lecture Series, Chapter 22, the Lymphatic System and Immunity. Now this chapter, to some degree, is a continuation of chapters 19, 20, and 21, which were blood, heart, and blood vessel. Now, in this story of chapter 22, we have two things going on. One is the lymphatic system, and the other is immunity. Now, chapter 22 is somewhat of a continuation of the story that we had with chapter 21 at the end of the AACVV. At the VV end, we talked about reabsorption. We said something about 85% of the fluids are reabsorbed at the VV side because of blood colloidal osmotic pressure, which is related to albumin. Well, this is exactly a continuation of that, because blood, as in arteries versus veins, not only are they parallel with each other, we also have a third aspect, which is the lymph. So when we talked about 85% of our fluids being reabsorbed at the VV end, the other 15 that we did not talk about is the drainage into the lymphatic system. So when it comes to the 100% of interstitial fluid, 85% will go in the reabsorption back into the bloodstream, whereas 15% of it will drain into the lymphatic system. And we'll see that a little bit more. Our first slide for chapter 22 starts off with this one. This slide indicates that we have pathogens that invade our body. And we have such things as viruses, bacteria, fungi, parasites. And each one of these items actually invades and attacks in a slightly different way. And therefore our response to these will be in a slightly different way as well. Our next slide starts off with the lymphatic system. Now let me backtrack for 1 second. And that is the slide I had a moment ago. One thing I need to mention before I move forward is that pathogens will elicit an immune response. That means that when we have germs in our body and our immune system notices that they are different from our self-antigens, so we have self-antigens and we have foreign antigens. And it's again a surface marker story that we're talking about. So when these invade our body, they have different surface markers, and we respond by creating an immune response to those surface markers. Okay, now let's continue. So here's what the lymphatic system does in general. It protects us against disease. It does it in several ways. One of the ways it deals with is environmental pathogens, germs that are in our environment. Then toxins, the toxins that we in our own body we build. So we have external toxins that come into us, and we also have toxins within our body as well. And then we have the abnormal cells such as cancers. So you could say that the lymphatic system plays a role in our defense, in our immune system, in our defense against germs and cancers. Then we have the next slide. Now, before I go further, I want you to notice the terminology at the top. Before we finish this chapter, or as we actually go through the chapter, I'm going to ask you to create a chart for me. The chart that we're creating is going to be called Level 1, Level 2, Level 3, Level 4. Now each of these levels has certain things within it as columns. So column 1, column 2, column 3, column 4. And each column is unique. And in this case, we're talking about the word specific. So what you're going to find is that we're going to call column 3 and column 4 of our 4-column chart we're going to create as specific. So column 3, column 4 will be specific. That means they go after a very specific pathogen. The slide says it well here. So anytime you see a specific defense, it is going after a specific pathogen. Levels 1 and levels 2, so level 1, level 2 are non-specific. They're basically just generic germ fighters that go out and hunt for stuff that looks foreign. Whereas levels 3 and 4 are specifically going after something. Now whenever you use the word specific, Notice it says right here the word immune. Well, by true strict definition, anytime you say immunity, immunity refers to a specific. So level 3, level 4 will be specific. Okay, next slide. Now here's what I was saying earlier. The immune system, we have our immunity. Notice it says it's the ability to resist infection and disease. All body cells and tissues involved produce immunity. So immunity, or the fighting of the germs, is all throughout the body, but it's the immune system of the lymphatic that we primarily key in as our immune system. But all body cells have some degree of immunity involved. Next slide. Here's what I said earlier about nonspecific. So as we're now making our chart along the way, we'll see that nonspecific refers to levels 1, levels 2. Notice it says block or attack any potential infection, cannot distinguish one attack from another. So it does not know if it's a specific or a non-specific. It just does what? Attacks anything that looks different. Different from what? The self-antigens. So anything that's not self is invaded. Whereas in the case of true immunity of levels 3 and 4 with the word specific, they know exactly what they're going after. They're going after a specific marker on a specific antigen or a specific germ. Next slide. Here we have the other organization, lymph. Now, lymph is very, very similar to plasma, but it does not have plasma proteins. Now, remember the story we said about the VV end and edema. If you somehow have leakage of plasma proteins into the interstitial fluid, and in this case leaking into lymph, fluids are going to go into lymph or into the interstitial, and you're going to have edema. So that's not good. So lymph is very similar to blood, very similar to plasma, but does not have the plasma proteins because the plasma proteins should stay in blood. Now, lymph vessels. So this time around in this chapter, when we talk about lymph vessels, or so-called blood in quotes vessels, we're really talking about lymph. Now, lymph is going to connect to the venous system. So one way or the other, your lymphatic system is connected to the venous system of your circulation. Now, I want to go ahead and point out that how do I move lymph, since there's no such thing as a heart for the lymph itself? Well, lymph moves because of many of the same things that we had for venous return, such as skeletal muscle pump. That actually is squeezing the lymph and moving it forward. The lymph, like veins, have one-way valves. So lymph, like veins, have one-way valves. And then the other is the respiratory pump that we spoke of as well. So much of what actually moves the venous blood back to the right atrium is also moving lymph towards the venous side as well. Then we have lymphoid tissues, such as your spleen that belongs to a lymphoid tissue, your lymph nodes, okay? That type of thing is number 3, the lymphoid tissues. Okay. Now, the last item, number 4 there, it says lymphocytes, phagocytes. Okay. Lymphocytes, of course, you already know belong to the level, belong to the lymphocytes category, when lymphocytes were what? Your things like B cells, T cells, those were your lymphocytes. Okay. Moving forward. Okay. Here's our picture. Again, notice that excluding the brain area, we see lymph nodes pretty much everywhere in the body. Okay, so we see lymph nodes pretty much head to toe everywhere in the body. And as we said with blood vessels, the blood vessels differ along the way. Well, the lymphatic vessels also differ along the way. Okay, so make sure you know the definition of lymph and what lymph is, and make sure you're comfortable with the lymphatic vessel story. We're going to go into that in a moment. Now, what does the lymph system do? Well, one function listed here, it says, is producing and maintaining and distributing lymphocytes. So the lymph system deals with production, maintenance, and distribution of lymphocytes. Okay. Now, where are these lymphocytes productions? In places like tonsils, spleen, thymus, red bone marrow. Okay, so this slide is important. So lymph tissues like tonsils, spleen, thymus, red bone marrow. Okay, what about the lymphocyte distribution? It's based upon what? It detects the problems and it travels into site of injury or infection. So it actually travels there by way of your blood or by way of your lymph. Now, to me, the lymph system is sort of a quarantine station. So we gave out numbers a moment ago. 85% was reabsorbed back into blood and 15% ended up in the lymph from your interstitial space. Well, that 15% is going to serve as a quarantine area. So when you have germs that are infecting your body and your lymph nodes are swollen because of an infection, you're going to find that that is going to quarantine the germs and then kill them off. So what would you expect to find inside of a lymph node? You would find red blood cells? No. White blood cells? Yes. So you would find macrophages, you would find B cells, you would find T cells, right? So B cells, T cells, macrophages are what you would find inside of a lymph node. Okay. They're there to kill off the germ once it's been quarantined. Next, we have this slide. From blood to interstitial fluid through capillaries. And notice, as I said earlier, we're going to have the lymphatic system meet up with who? The venous blood. So that's what this slide is now showing you, the same thing. So it returns to the venous blood through the lymphatic vessels. Then we have the bottom half. It says again, blood plasma to lymph and back to the venous system. All right? So we're really doing what? Circulating the fluids, but still returning back to the venous side of our blood. And yes, we are transporting things like hormones, nutrients, and waste. Okay, so we are transporting things like hormones, nutrients, and waste. To me, the lymphatic system is sort of like a drainage system, and a lot of people use that term, that it's a drainage system. Okay? So the lymphatic system is parallel with the blood system, and it does return back to the venous side of our circulation. Okay, next slide. Okay, this slide, don't care too much about this slide, just do understand that lymph vessels carry lymph. Other than that, I don't care for this slide. Next slide. This one I do care for. Make sure you know the characteristics of capillaries that are lymphatic capillaries versus blood capillaries. So lymphatic capillaries versus blood capillaries. One, lymphatic capillaries are larger. They have thinner walls, start as pockets rather than tubes, and are flat typically, or irregular shaped. Okay. So these are the differences between blood capillaries and lymphatic capillaries. Okay, do not confuse the two. Next, we have a picture version of what we just said. Do you now see what I mean by everything paralleling each other? So we have our picture here showing us the artery and arterial side of our story. Then here's the interstitial space. We had words like precapillary sphincters and all that before. And then we have our vein and venule system here. And notice it is related to, neighboring to the lymphatic system. Okay. So the lymphatic system is our drainage. It will take up 15%, whereas this side will pick up 85%. So the VV side will pick up 85%. The interstitial fluid space will pick up the remaining 15%. But in the end, they both come back to the venous system. Okay, so this slide is worth studying. Next slide. This slide shows you the flow and what happens. So we have lymph flow shown here coming from the interstitial fluid. We have our little lymphocyte there. We have our blood circulation nearby. Interstitial fluid again dumping into the lymph, going to larger lymphatics. So your lymph system is really having a lot of drainage role, as well as whatever's dirty, whatever germs you've got, they're going to drain from the interstitial fluid into the lymph fluid. And that's why your immune system is ready to go in the lymphatic system. You would expect that, right? You would expect to have your immune system right next to your drainage area. Next slide. Okay, I've already mentioned this. I'm going to go quickly through this slide. We said that the lymphatic capillaries have what? One-way valves. Okay? That means that whatever comes in is moving in one direction. So it allows fluids, solids, viruses, bacteria to go right into from the cell space. And notice what it says. Prevents return back to the intercell space. So if germs are drained from your interstitial fluid space, they can't go back to the cell space anymore. They're now in the lymphatic. And that's where your immune system is going to quarantine and kill off these things if it possibly can. Next slide. Lymph flow. Lymph flow, we've already talked about. It goes from capillaries to larger lymph vessels. So from capillaries to larger lymph vessels containing one-way valves. Notice what it says here. Lymphatic vessels travel with veins. It's almost like a parallel circuit with the veins, and eventually they join the venous system. I keep saying that because we'll see it in a moment. Then we have lacteals. Now this is a different role. When you go to eat at Golden Arches and you eat your food that contains carbohydrates and proteins, well, the carbohydrates are going to be broken down into glucose and small monomers, whereas the proteins are going to be broken down into amino acids. Those two items will actually be absorbed into the bloodstream in your intestines. That is not true for the fats. Your triglycerides, your fats that you eat, they are going to get absorbed into the lacteals. So in your GI tract, in your small intestine, you have things called villus or villi. And the villus or villi actually have blood vessels and lymph within them. And the blood vessels will absorb the amino acids and the small glucose monomers, whereas the lymph will absorb the fats, the triglycerides. And in this case, that lymph that we have in the villus of the small intestine, they're called lacteals. So the author says lacteals transport lipids from the digestive tract. So the lymph that you see within the villus of the small intestine is really called a lacteal. So you're actually absorbing dietary fats through the lacteals in your small intestine. Okay, next slide. Here's a picture of how everything relates, very similar to the previous one. But notice, veins and lymph are very much related to each other. Now in some cases, as shown here, you actually see the one-way valve. So that's actually your one-way valve making sure everything goes in one direction towards what? And we've said that many times, towards the venous system. Okay, towards the venous system. Here's an enlarged view of the valve, making sure that fluids only go in one direction. Okay. And it says it right here, permits movement of fluid only in one direction. So it's a unidirectional way of making fluids move. Again, the goal is to get it back to the circulation. Next slide. Okay. Now this slide talks about superficial lymphatics, deep lymphatics. Be aware that they're pretty much where? Everywhere. One of the other slides said that the lymphatic system is found in pretty much everything. So you have lymphatic vessels in your skin, your mucous membrane, your serous membrane, pretty much everywhere. The reason for that is wherever the bloodstream will be, that is where the lymphatic will be. And that's a shortcut way of remembering that. So wherever you find blood system, you find the lymphatic system. So the only places that we saw earlier in 2401 that we had a poor blood supply was cartilage. Right? Cartilage had a poor blood supply. So you would expect very little lymphatic vessels there too. So you pretty much see them wherever you see a blood vessel network. Skin is good, rich in blood vessels. So is mucous membrane, so is serous membrane. Next slide. This slide I want you to look at. What's more important about this are these last two. So on this slide, the biggest importance is the last two, the thoracic duct and the right lymphatic duct. So the lymphatic vessels have a flow chart, and one of the things they'll end with is the thoracic duct and the right lymphatic duct. And there's a better picture coming up, but make sure you know the thoracic duct and the right lymphatic duct. Next slide. Okay. Don't care too much about this slide. Next slide. This slide's important because this slide says thoracic duct, and as I said a moment ago, it's important. Collects lymph from these places. So the left bronchiomediastinal trunk, left subclavian trunk, left jugular trunk. And then notice it empties where? Into the left subclavian vein. That is very, very important. Okay. So the thoracic duct will empty into the left subclavian vein. This is where your lymphatic system joins the circulatory system. We've been harping on that for the last few slides, saying it joins the venous return, venous return. Well, what we're saying is it joins into the subclavian vein, the left subclavian vein. Okay, next slide. All right, here we go. The right lymphatic duct will have the right jugular trunk, right subclavian trunk, right bronchomediastinal trunk, and it empties into what? The right subclavian vein. So you can see that we have two passages. Stuff on the right side of the body will end up where? In the right subclavian vein. Stuff on the left side of the body will end up in the left subclavian vein. And where is that subclavian vein? In the shoulder region. Now, to make sure you understand that, we have a picture. And that is this picture. This picture shows you, on this side, we see the left jugular trunk. Right, we see all that there. And then this is, of course, the left subclavian vein. So this is where everything meets up. And on the left-hand side, you had your thoracic duct mentioned. There is no thoracic duct on the right-hand side. So the right-hand side, again, I'm talking about the patient's right. The author shows it very well here. So the patient's right has the drainage of the right lymphatic duct. On the left, the patient's left, we have the drainage of the thoracic duct. So that's where we have an asymmetry story. Just like we had the asymmetry with the aorta of the heart, we have an asymmetry here as well. So the person's left has a thoracic duct. The person's right has a right lymphatic duct. Okay, so be aware of that story. Okay, so next slide. We've already talked about that. Be aware of those two. Here's the enlarged view of the patient's right and the enlarged view of the patient's left. Okay, any questions on those? Okay, continuing, the next slide. Now we have the lymphedema story. If you, for some reason, plug up that number of 15% that we keep talking about draining into the lymphatic system, we're going we're going to get severe edema. So severe edema. This often happens when you have a condition called elephantiasis. It's a parasitical infection where the worm goes in when you're walking barefoot in a pond. The worm starts growing, burrows into your skin, very, very tiny, starts growing into your blood vessels, and it grows into a long worm that blocks up the lymphatic vessels. In that case, the 15% of the fluids that normally would drain into the lymph backs up into the cell space, and you get a big fat leg that looks like an elephant's leg, hence elephantiasis. Now, it does interfere with the immune system. It's not going to be life-threatening, but if you do get an infection, that infection will have a free reign of that area because now it's going to gobble up, eat up, infect whatever it is because it's now finding a nice home where the immune system can't get to it. Okay? So it interferes with the immune system. Then the bottom says lymphocytes. Now you learned this already in your chapter 18 on the blood, or 19 on the blood. Make up 20 to 30% of circulating leukocytes, right? And what kind are lymphocytes? Lymphocytes are agranulocytes. Remember that from before. Okay, so the lymphocytes. And we have two types, B lymphocytes, T lymphocytes. And as we said, in this chapter, we're going to be making a chart. Well, the B lymphocytes will belong to level 3. So B lymphocytes will belong to level 3. T lymphocytes will belong to level 4. So we're making our chart as we go along. So B lymphocytes level 3, T lymphocytes level 4. Okay, now we come to this slide. So the types of lymphocytes. A moment ago, I told you T cells or T lymphocytes. I'll quickly talk about that and then we'll move forward. The T cells or T lymphocytes are our level 4. And as you by now know, that level 4 is specific. Now, where are T cells made? T cells are made in the bone marrow. So T cells are made in the bone marrow. But where do they mature? They mature in the thymus. So T cells made in the bone marrow but mature in the thymus. Now the unique thing about the lymphocytes for T cells, the T cells, their job is to deal with extra and a more of a intracellular role. Now, let me give you an example of that. So a little bit of extra but more of an internal role. And the internal role is for viruses and cancers. So T cells will deal with the internal with viruses and cancers. So once a cell is infected internally and the DNA is screwed up, it's the T cell's job to go in and kill off the virus-infected cells such as cancers. Yes, AIDS is a cancer. It is a contagious cancer, but it's still a cancer. So T cells go with internal aspects of infection. B cells now. B cells are also made in the bone marrow. And now we're talking about level 3. So B cells, level 3, also made in the bone marrow, but they mature in the blood. So B cells made in the bone marrow, but mature in the blood. Now, B cells are truly for extracellular, typically extracellular bacteria infections. So germs that are on the outside of the cell. That's what they're for. Okay, so germs that are on the outside of the cell. Okay, any questions on that? Okay, next. For a moment, we're going to hold off on natural killer cells. They'll come back in a moment. Okay, so we have B cells, T cells. B cells are level 3, T cells are level 4. B cells for extracellular and T cells are for intracellular, such as viruses and cancers. Okay, and we'll come back to NK later. Next slide. T cells. They make up most of the circulating lymphocytes, 80%. They come in different forms. Now, each of these forms that you see here, cytotoxic, we're going to describe each one. The cytotoxic cells, their job is not to comfort the cell, to sort of help the cell along, to redo the cell. No, their job is to kill the cell. So cytotoxic cells are those killer cells that we typically call that destroy the cell. They look at a cell as a recidivist. It's a term in law that says, hey, the kid is not fixable, and the kid, in this case, the cell needs to be destroyed or locked up, something like that. So in this example, the cytotoxic cell's job is to kill the cell. It does not like to rehabilitate it, it just kills the cell. So cytotoxic cells will kill that, especially who? Cancer cells. Remember, we're in the category of T cells. So cytotoxic cells are killer cells. Their job is to just destroy the cell. Next, memory T cells. Memory T cells are with that line of level 3, level 4, that word specific. And we said that anytime we say level 3, level 4, and say specific, we have the word immunity. Well, immunity refers to memory. Okay, so it refers to memory. And therefore, if you are infected once with that same exact germ, having the exact same antigen surface marker that you responded to the first time around, it will remember that and it will be ready to fight the second infection quicker. And that's why we have memory T cells. So memory is only found whenever we say specific. Next, helper cells or helper T cells. The helper T cells are the ones that play a supportive role. Their job is to actually link the levels 1, 2, 3, 4 that we're making in the chart. There's a nice picture later in our chapter showing you that the helper T cell is the one that sort of connects Nemesh's level 1, 2, 3, 4 and puts them all together. So it's the interconnecting of the different levels. That means if you have AIDS, HIV, and you have a depressed amount of helper T cells, what happens to your overall immune system, since these play the main role of joining levels 1, 2, 3, and 4 together to fight the infection? You can't. So if you have a sudden drop in helper T cells, your overall immune system is starting to fall, starting to fail. Okay, suppressor T cells, not much to say there. Okay, here's the author's version of the same thing. So we've already said cytotoxic T cells. It does what? Kills infected cells that are infected by viruses or cancers. Now this line right here is important. It says T cells play a role called cell-mediated immunity. That is going with level 4. So when T cells, level 4, we call it cell-mediated immunity. We already said that memory goes with what? Immunity. Again, that means it must be specific. Therefore, we're talking about level 3 or 4. In this case, we're talking about level 4. Again, it has to be based upon the exact same antigen as before. Okay, next down here it says helper T cells. And I told you that it plays a role in joining levels 1, 2, 3, and 4. The author mentions here B cells because it is connecting level 3 B cells with level 4 T cells. Next slide. Okay, I didn't say too much about suppressor, and I didn't mention too much about regulatory. Okay, so I'm not concerned too much about these two slides. Next. B cells down here. Okay, so we're now moving to the bottom half of this slide, the B cells. Now B cells are unique, so we're now all of a sudden switching to level 3 now. So level 3. Well, level 3 is still what? Specific, right? So level 3 is still specific. That means we're still using the word immunity. Now here's what's unique about level 3. We've already said that B cells are extracellular stuff, so bacteria that are outside. But in order for B cells to do their job, they have to change into plasma cells. So B cells will produce plasma cells. Now these plasma cells will in turn produce antibodies. So B cells turn into plasma cells produce antibodies. And in other languages or in other countries, they often refer to antibodies as immunoglobulins. Now these antibodies are made by specific plasma cells. That means it's not all for one. Each plasma cell produces a unique antibody fighting off a unique antigen or a surface marker. So it's not all fits all. One antibody fits one type of germ. That's what I'm getting at. So specific B cells, specific plasma cells producing specific antibodies. Okay, next slide. Now let's go to antigen. Now the definition provided here is okay, but like mine's a little bit better. What is an antigen? Anything that elicits an immune response. That is what an antigen is. Anything that elicits an immune response. Now we said a moment ago that antibodies go with B cells. So your book has a term for that. So B cells are considered antibody-mediated immunity because they're making a specific antibody to a specific target antigen. So a specific antibody to a specific target antigen, and that's called antibody-mediated immunity. Next slide. We've already talked about the top. We're now going to mention natural killer. This is what I skipped a moment ago. So natural killer cells responsible for immunological surveillance. And they're the ones that fight off virus-infected cells and cancer cells, very similar to the cytotoxic cells. The only catch is this: natural killer cells belong to level 1. So they're not specific. So natural killer cells are not specific. Whereas cytotoxic, those special type of T cells, they are specific. So therefore they would belong to the level 4. So let me say that one more time so we have it clear. Natural killer cells are level 1. Whereas cytotoxic, the T cells that do that, they're level 4. So big difference between them. Very similar names, but different role. So natural killer cells are level 1, whereas cytotoxic, they are level 4. Okay, now we're going to look at this slide. This slide is a picture version of all the various forms of T cells. This is the one that I keep saying is a killer cell, but it's level 4. Again, it too does what? Fights germs infected by viruses, but this is part of level 4. Then we have helper cells, suppressor cells, and memory cells. And they can be converted to any of these forms. Next slide. Here's the B cell. And the B cells make plasma cells, and the plasma cells would make antibodies not shown in this picture. And then we have a separate category called natural killer cells. And these are not really true specific. Next slide. Okay. Tissues maintain different T cell, B cell populations. And now the unique thing about the T cells, B cells, because they are memory, they are immunity, they are specific, they can survive for many years. So your vaccination role, for example, once you're vaccinated, they last for many years. This is why. Because you make B cells or T cells with memory, therefore they can last for many, many years. We're talking about decades even, like the booster shot for tetanus can last a decade. Okay? So anytime you talk about B cells, T cells, remember, they can survive for many years, decades. Okay, next. Okay, now before I move to the next slide, let's make sure we're clear on this comment. When I said decades a moment ago for B cells, T cells, I'm again talking about level 3, level 4. That is not true for level 1 and 2, which are non-specific, and therefore they don't do these things. Okay, next slide. This is a general term that we had before, lymphopoiesis. Just like erythropoiesis, hematopoiesis, it simply means in this case, how do I make lymphocytes? Mostly we make them where? In the bone marrow. Okay, so mostly in the bone marrow. For T cells, they mature in the thymus. And then we have others like the spleen. Like the spleen. Now this bottom portion here, you actually saw in the blood chapter. So this is a review of that. You learned the granddaddy or grandma of all of the blood cell types, whether it be red blood cells, white blood cells, platelets, whatnot. They came from hemocytoblast. Well, the hemocytoblast had a branch on that chart that we saw before in the other chapters. And in that chapter, they called it the lymphoid stem. So the lymphoid stem had lymphoblast, and lymphoblast produced the lymphocytes. And that's just a review from a chart we saw in chapter 19, I believe. Okay, next slide. I don't care for this slide. However, I do care for this picture. So this is a shortened version of the picture we had in chapter 19. So this is a shortened version of it. Now here's what I want to get at. Do you see this word right here? Interleukin. Now, when we were in the chapter 19 on blood, and I mentioned that chart, and that chart, and let me go ahead and get to that page myself so I can refer you to that page. But in chapter 19, we had a chart that looked like page 659. So page 659. And I mentioned a word there. What allows us to go from one level to another on page 659's chart? Well, it was called cytokines. And what are cytokines? Cytokines are an umbrella of chemicals that tell a cell how to react to its environment. Well, one of the cytokines you now see here is called interleukins. So interleukins are chemicals that tell a cell how to react to its environment. So interleukins would be an example of cytokines. Okay, so interleukin is an example of cytokines. Okay, next slide. This is your next slide. Now this slide I'm not too concerned with, but I do want you to understand that how do I get a B cell to do something? How do I get a T cell to do something? Typically we're talking about some form of chemical message, like in this case, cytokines. And notice it now does say what? Cytokines. And interleukin is an example of one. And we're going to see a couple more. So interleukin is an example. Okay, next slide. Now, this slide right here, it says in the last part, is essential to immune system function. That means if you lose level 3, level 4, you're in big trouble when it comes to your immune system. I'll mention a kid named David, the Bubble Boy. In the late 70s, there was a movie with John Travolta about David the Bubble Boy. And the real David the Bubble Boy had an immune system problem called SCID, Severe Combined Immune Deficiency, where his level 3 B cells and level 4 T cells were not working. And he was solely relying on level 1, level 2. And that wasn't good. So he quickly soon died thereafter. Okay, so he could not be exposed to the everyday germs that we had around. Okay, this picture or this slide, don't really care for. This picture, I want you to be aware of what a lymph node looks like. This is one shown here in the intestinal area. Okay, so be aware of that lymph nodes and lymph nodules are pretty much everywhere. And this is one in the intestinal area. Why would you have it in the intestinal area? Because you don't know what you're eating, right? You've got all kinds of germs that you're eating. How kids eat mud at the playground, right? So you have germs everywhere. So you've got a lot of immune system in your gut. Much of the reasons of getting sick are often things that you ingested. In your throat, your tonsils, and that's the next one I'm on. In your throat, the tonsils, same thing. Their job is to do what? Keep you safe by trapping the germs that you put into your mouth. Okay, next slide. Now, where do I find these lymph nodes or lymph nodules? I find them as lymph nodes, find them in the spleen, find them in the respiratory, that's why they're in the mouth, in the digestive, in the urinary, reproductive. Now, what's the best way to remember this? Anywhere and everywhere, especially where you have what? A connection with the blood. Let's take an example of this one. Spleen is connected to your blood. It has a rich blood supply. Respiratory, connected to your blood. Digestion, connected to your blood. Urinary, connected to your blood. Reproductive, connected to your blood. That's why things work down there. Okay, so you see, anytime you have a nice connection with your blood, you're going to have lymph nodes and your lymph system there. Okay, next slide. This slide is worth copying down. I would definitely read what these two are, and let me give you a page number. Page 773. So page 773, you want to read completely, the entire page. So page 773, read the entire page, including the clinical box. So page 773, the entire page. And it does talk about MALT, the mucosa-associated lymphoid tissue. Okay. Next slide. The tonsils. I'm not too stressed about the tonsils, but I do want you to know what their role is. Next slide. Okay. We've already seen this. Next slide. This slide is also mentioned on page 773, so we're going to pass that up. Same is true for this, so I'm going to pass that up too. But I do want you to study this picture. Okay, so this picture is important. So here we have a lymph node, and we have blood going in, we have stuff leaving. And that's another view of it. Next slide. Now, this one is important to keep in mind. I do want you to have a flow of the lymph flow through the lymph node. So have a general flow. Now, if this does not help, go back to the picture I showed a moment ago. So this picture actually goes with that slide. So have a general feel for the flow through there. Next slide. What does a lymph node do? And I've used the word quarantine several times. The object of the game is to filter, and a filter is very much like a quarantine. What does it do? It removes debris from the interstitial fluid space. It removes pathogens by isolating them. And then it also kills up and destroys whatever antigens you got floating around in there. So its job is to find the antigens, because what does an antigen do? It elicits an immune response. So our job is to break that down. Okay. Now, how does a B cell or T cell or anything that for that matter, how does a cell recognize an antigen. When it comes to recognizing a germ, we have what's called antigen presentation. Now, this slide by the author is sort of jumping ahead of the game. Now, I've already told you that level 3 B cells, level 4 T cells are lymphocytes. What this slide is trying to tell you is that we are going to present the germ to them. Now, what this slide does not say is who is doing the presenting. Well, that is level 2. So in your chart, you should have level 2 with macrophage, monocyte, and phagocytosis. And that would be level 2. Now remember, level 2 is not specific. Okay, so level 2 is not specific. So level 2, macrophages, monocytes, and phagocytosis, they are going to kill the germ, destroy it, and then present the antigens to level 3 or level 4. And that is how these levels are interconnected. So level 2, your macrophages are going to chew up the germ and then present the antigen part to level 2, to level 3 or level 4. So where it says presented to, it's really the level 2 that's doing the presenting, either to level 3 B cells or level 4 T cells. That's not mentioned here, but that is what it's getting at. Okay, I really don't care too much about that slide. On this slide, I only care for the bottom part, where we have the lymphadenopathy. So you have an enlarged lymph node. Several reasons for that: an infection, a hormone problem, or cancer. So this can be a very dramatic issue. Now, one of the things you need to realize for the lymphatic system is if you screw up the lymphatic system really badly, if it's really poorly off, you're going to have a lot of dire consequences, not only as an infection issue, but as an endocrine issue. Many of those medicines you see on TV, they'll often say that if you are fighting rheumatoid arthritis and you take this medicine, it can cause problems with blood clotting, problems with infections. Okay? Do not be taking such a medicine. Well, that's because if you mess up with the immune system, you can cause problems with the hormone system as well. They're connected. Okay, next slide is the thymus. For this slide, I want you to know where it's located. So it's located in the mediastinum. I want you to know that it atrophies after puberty. It means it's going to cut down on the effectiveness of the immune system. That's why you're strongest before puberty, because you have that protection of the thymus. Now remember, T cells are made in the bone marrow, but they mature in the thymus. Next slide. This slide I don't care too much about, but I do want you to be aware of it, and that is the same for the next slide. So this slide, and this slide, I want you to be aware of them. Here's the picture next showing you the thymus and where it's located. Now I do want you to understand this picture showing you the parts. Remember there was a slide a moment ago showing you the names of these parts. So be aware, familiar with the names of the parts mentioned on the 3 slides before. So be aware of the names of these terms 3 slides back. Same is true here. This was mentioned on the slide before as well. Okay, now that takes care of the thymus. The same is true for the spleen. Now you have come across the spleen before in the blood chapter of chapter 19. In that chapter you learned that the liver and the spleen's job is to destroy the red blood cell after 120 days. Well, in addition, the spleen plays a role in activating B cells. So spleen will play a role in activating B cells. That is item number 3 on this slide. So it activates B cells. Now the spleen will also play a role in working with the macrophages, and those are the ones I mentioned that go with level 2. Okay. This slide, So the slide before, this slide, they're all related to the positioning of the spleen, what the spleen looks like. So this would be a lab item. And within the micrograph of the spleen, here's what it looks like. So these terms that you see on the right-hand margin, these terms were on the slides that I just passed up. So be generally aware of terms like red pulp, white pulp. Be aware of what happens in the red pulp. And again, remember, red pulp goes with macrophages. So macrophages. Don't care too much about that one. Then the spleen, the micrograph of the spleen, and now I'm on my next slide. Okay. So the spleen plays a role in the production of the macrophages and the activation of the B cell. Okay. So macrophages belong to level 2. That's right. Next slide. Here's what the spleen does. I've already talked about it, so we can move on. Okay, this is a new slide now. Now on this slide, we have a big issue. This is where our chart is now going to come into play. Anytime we talked about nonspecific, I mentioned that nonspecific will be level 1, level 2. So level 1, level 2. Then specific will be level 3, level 4. So we're using that same 4-level system that I've come up with there. Okay, so non-specific, specific. Let's move to the next slide. As I move forward from now on, what we're going to be doing is putting all of this into our 4-level chart. Here's the next slide. So this slide right here indicates non-specific. Notice it goes against any type of invading agent. It does not matter what it is. It's not going after a specific antigen. The only ones that go after a specific antigen are the ones down here wherever we say immunity. So immunity, specific, memory, all that goes down here. So if you want long-term immunity, it's got to be levels 3 and 4 mentioned down here. If you want just a quick infection control, that would be non-specific levels 1 and 2. Okay, next slide. Now, this slide right here is a mixture of my level 1 and my level 2. So here's how we're going to break this one down. Item number 1 is physical barrier. That will be my level 1. Now under physical barrier, we're going to put a slash and call it physical and chemical barriers. So physical, chemical. I used to call it mechanical and chemical, same thing. So physical slash chemical barriers. And that's going to be my level 1. Now what does that mean? It means that I will have things like nose hairs, okay? Like having a longer piece for urethra versus female, so male versus female urethra. Okay? Anything that is a barrier, like nose hair, earwax, all of these things are barriers for germs. Okay? So physical barriers, chemical barriers. The act of urination, that would be what? A physical barrier. It's a mechanical thing. Urination does what? Gets stuff out. So number 1 is under my number 1, level 1. Okay. Now, number 2 here, phagocytes. That is actually level 2 for me. So my level 2. Now that goes with monocytes and macrophages. So monocytes, macrophages will be my level 2. So let's make sure we're together. So what's this one right here? Physical barriers. That's my level 1. This one here is my level 2. Now we're going to look at level 2 a little bit more before we go further. We said phagocytes. when we said macrophages. And we also said earlier that macrophages or monocytes are going to help us with the presentation of the germ after it's been chewed up. It's going to present the germ to level 3 and level 4, the B cells and T cells. Well, these macrophages come in two varieties. We have the free macrophages. Those free ones are just moving around in your blood from point A to point B. We also have fixed macrophages. Now, fixed macrophages are like the Houston Police Department having those little substations in unique areas of Houston. Areas that have high crime, if they have a substation right there, what can they do? They can actually monitor the crime that's in that area and be more responsive to the community. Well, that's exactly what fixed macrophages are like. Now, let me give you an example of a few fixed macrophages. One is called Kupffer cells. Now, Kupffer cells are your liver's fixed macrophages. Why would you want to have fixed macrophages in the liver? Because the liver's job is to clean up your blood. And if your blood is dirty and it's got germs in it, the liver's got to cope with that. So you have fixed macrophages in the liver called Kupffer cells. You also have them in the intestinal area called Peyer's patches. So Peyer's patches are found in the intestinal area and they are fixed macrophages. In the lungs, which is our upcoming chapter, in the lungs we have them called dust cells, and some people call them alveolar macrophages. So as you can see, we have fixed macrophages in key regions where infections are likely to happen. Even in your skin, you had fixed macrophages. Do you remember them from 2401? In 2401, you learned about Langerhans cells or dendritic cells. And those dendritic or Langerhans cells were your fixed macrophages of your skin. In essence, we have fixed macrophages for anywhere we have a key point of entry into the body. You get things on your skin, that's a fixed macrophage location. You get them in your lungs because you inhaled something. You ate something, so it's in your intestinal region, and then in your liver because it deals with a lot of that. So does that make sense? Okay, next, we now move to number 3 here. Now, number 3 was your natural killer cells. And remember, I said that they actually are not specific. Now, this belongs to my level 1. So the immunological surveillance there is natural killer cell, and it actually belongs to my level 1. All right, now we move to interferon. Interferon is also my level 1. Now, what are interferons? Interferons are chemicals that come from a virus-infected cell. So if a cell is infected with a virus, that infected cell puts out a chemical message bulletin. And that chemical message bulletin is called interferon. And it tells neighboring cells, hey, I'm infected with a virus or a germ or whatever, and this is how I'm coping with it. And if you should by chance come down with it, how are you going to deal with it? It's a memo to other cells. Now, as a result, it belongs to our umbrella again of cytokines. Remember what cytokines were. Cytokines are a group of chemicals that tell a cell how to react to its environment. Just like interleukins were earlier, interferons also tell a cell how to react to its environment. So interferons come from virus-infected cells telling other cells how to deal with it. So it too is a cytokine, and it belongs to my level 1. Okay, we move on to the next one, which is complement. Now, complement is a protein. Okay, so it's going to be a protein. And complement's job is to work with level 2. So that means I'm still on level 1 here. So complement is a level 1, but it's going to work with level 2 And level 2, of course, were the macrophages. So having a chart, as I've indicated, makes much more sense. Now, what is complement? It is a protein that you coat the germ with. So if you have a germ and you want to make that germ more visible, what do? You coat it with this complement protein. It makes it more visible. Now, of course, you're making it more visible for who? For level 2, the macrophages. A good example that I give is if you have kids who are learning how to drive, paint their car, school bus, yellow. If your kid's car is school bus yellow, believe me, everyone will see it coming down the street. Same way here. If you have a germ and you coat it and cover it up with this complement protein, the macrophages will identify it more readily. So that's what a complement is, and it belongs to my level 1. It's not specific. Okay? And we'll see more of that in a moment. Then we have inflammation. Inflammation is a level 1. Its job is to bring more what? More white blood cells to that area, basically to have a more stronger immune response. So anytime you have inflammation, it's going to make your immune response stronger. Number 7 is fever. Fever again is number 1 on my column. So level 1 for fever, just like inflammation's level 1, fever is 2. Now fever is good and bad. If you raise your body temperature too high with a fever, you might denature key proteins for other functions, and that's why fevers aren't good. But a fever itself's job is to do what? Raise the body temperature so you can kill off the germ, because germs have a narrow range of survival. Okay. So that's why the body tries to have a fever. Again, that's level 1, 2. Okay, so now you should have a chart forming with level 1, 2, 3, and 4. Next slide. Now I'm going to go quickly through these because this is a rehash of what I just went over. So we have physical barriers or chemical barriers like your nose hairs, your earwax, your urination, defecation process. It just keeps things outside the body. Phagocytosis, we've already talked about. That's my level 2. Then we talk about natural killer cells that are always looking out. Again, they are non-specific, so level 1. Okay, next slide. Interferons, I already told you that they are coming from who? Virus-infected cells. So interferons are coming from virus-infected cells. Okay. It's a response to that. Notice it says, does not or do not kill viruses. Okay, so these interferons will not kill the germ, will not kill the virus. They'll just make it so that we detect that the virus is inside of the cell. Okay, so that's what interferon. This is a chemical message saying I'm infected with a virus. Other normal cells, be aware that I've got a virus in me, and be aware that maybe you can boost up your own immune system somehow. Then we have complement. Assist the antibodies in the destruction of the pathogens because it coats the germ, makes the germ more visible, more palatable. Again, complement will help level 2 with phagocytosis. Okay, next slide. We've already talked about this, and we've already talked about fever as well. Next slide. This slide shows you a picture version of pretty much everything we've said. I've mentioned fixed macrophages and free macrophages already. Okay, so this is an idea of how that works. Picture version of the stuff we've talked about. Then, next slide, continues complement. Now I want to show this picture since I don't have a way of doing it otherwise. Complement are these proteins. And what does it do? It coats the germ. Now it says it lyses the germ, but not really here. Remember I said that coating the germ with complement is going to help level 2. And level 2 macrophages are the ones who are going to kill the germ. So this picture is sort of jumping the gun a little bit, and it really belongs to level 2. So this part is level 1, that part would be level 2 by way of using macrophages. But complement is my level 1, and the destruction by way of macrophages is my level 2. Okay, and the rest we've already talked about. Okay. I'm going to pass up this slide. It is again a rehash of what I've gone over. So skin, hair, flushing like urination, you're sneezing with the mucus coming out of you, you're sweating. All of that are non-physical, just barriers. Okay, so stomach acid. Remember I said physical barriers to me is mechanical and chemical. So stomach acid, enzymes, they're all barriers. Okay, now, starting with this slide, you know that I'm now moving to which level on my chart. By saying microphages or macrophages, I'm really talking about level 2 now. So this is level 2 that we are discussing. Again, level 2. Remember this phrase I gave you for level 2. Monocyte breaking down into macrophages, because monocytes make macrophages. Okay. So we're now talking about level 2. Now what's going to happen? You're going to activate these macrophages. Okay. Now, this slide here is a summary of what that macrophage will do. It will engulf the pathogen, destroy it using lysosome enzymes, those digestive enzymes you learned in 2401, and then it's going to destroy it. But of course, what did I say it'll do with the antigen it finds on the germ? It will save that antigen because it's later going to present that antigen to level 3 and level 4. Okay. So this slide is only telling you that it's going to break it down. Now, how does this even recognize the germ? How does that macrophage recognize the germ? Well, there's a term for it, and that term is called chemotaxis. So it's going to identify the germ by way of chemotaxis. It's going to recognize the chemical signature of that germ. So chemotaxis. Okay, so that's how it's going to recognize it. And once it chews it up, breaks it apart, it's going to present that antigen to either level 2 or level 3. Okay, next slide. I've already talked about this. I already mentioned the free macrophages, and I've also mentioned the fixed macrophages. And I gave you examples of fixed macrophages already, like the Kupffer cells of the liver, the Peyer's patches of the intestinal area, and the dust cells or alveolar macrophages of the lungs. Next slide. Here's my word if you had trouble with it. Kupffer cells. Kupffer cells were with the liver. So if you needed a spelling, there it is. So Kupffer cells went with the liver. Then we had alveolar macrophages, and I called them a moment ago dust cells. That's the other one. Okay, next slide. Okay, now I already mentioned this, so I already jumped the gun a little here too. I said that phagocytosis involves macrophages. What do macrophages do? They follow the germ through the capillary. So just as a germ can leave the capillary and go into the cell space, the macrophage can too. It follows it into the cell space, and that is called emigration. And you see the term in parentheses, emigration. Likewise, as I said, how will I recognize that germ? By chemotaxis, and I mentioned that already, chemotaxis. So it's like a chemical signature that I follow. and it recognizes the germ and then attaches it to it. So chemotaxis. Then once I attach to the germ, that is called adhesion. So chemotaxis, then comes adhesion. Next slide. We've already talked about this. I don't want to use this slide, but I do want to use a picture that most books have, and I'll be using that in a moment. Okay. Next slide. It is a good idea to read this slide, but I'm going to use the picture version instead because I like the picture version in most books. So here's the slide and now the picture version. Here's what happens to the NK cells. Again, remember, they are not specific. Here's an example. Here's an NK cell finding an abnormal cell. So an NK cell, natural killer cell, finding it. Remember we said chemotaxis and then adhesion. What will happen next? It's going to align with it, lock up with it. Then it's going to secrete perforin. And what will that do? It'll go in and start etching away at the abnormal cell. And that's what they're going to show on the next slide. So there you go. It's etching away at the membrane, and then the membrane will bust and you'll have a lysis of the abnormal cell. Now keep in mind, all this is doing is just killing the cell. It is not killing the virus inside of it. Okay, so it's just splicing up the cell. Why would you want to do that? Because if you already have a virus-infected cell and you let it stay inside that cell, the longer it stays inside that host cell, the more viruses it can make. But if you destroy the cell, the viruses already made have nothing more to do, right? They can't go anywhere. They can't make any more. Okay. So that's why you want to bust that cell. Next slide. We already said this. NK cells are for cancer cells. So cancer cells. And remember, it does not kill. So it does not kill the virus. Okay? It only kills the cell that has the virus. Okay? So it only kills the cell that has the virus. Okay? Again, this is a non-specific thing. Okay? Next slide. Let me make sure we're clear. See how it says it up here? Non-specific. NK. This is still my level 1. Allows the NK cells to identify and destroy them, but it does not kill the virus. It just kills the cell that the virus is living in. Next slide. Interferon. We've already spoke of interferon. We said that interferon was a cytokine. It allows a cell to react. Okay. So interferon is a cytokine. We've discussed it. Next slide. We have different kinds of interferons, alpha, beta, and gamma. This slide is worthy to keep in mind. So alpha, beta, and gamma. And this slide is worthy to keep in mind. It goes with the next slide. So here's your alpha interferon, beta interferon, and gamma interferon. Okay, and these slides are worthy to keep in mind. Next slide. Complement. I've already mentioned what complement is. It is a protein that covers the germ. So it's a protein that covers the germ. Now this slide is going to break off into 2 pathways. The classical pathway and the alternative pathway. So we're going to see those in a moment. Now the best way to understand this slide is to look at page 783 with me. So page 783. And I'll be there as we go along as well. So again, what is a complement so far? Is a protein covering that covers the germ, right? And it makes it more susceptible and more visible to levels 2, 3, and 4. Okay? Again, it belongs to my level 1 because it's not specific. Okay, now next slide. Here's the classical pathway, the FAST method, where the antibody molecule attached to the antigen. So we have an antibody attached to the antigen itself on the germ. The bound protein acts as an enzyme and it creates a cascading effect. Okay, that's why I said the slides are not that great. The pictures will be better. So here's the picture that goes with the classical pathway. Here's the classical. Here's our germ, in this case a bacteria. And what are we doing? We have our antibodies on the germ. And here's our example here. So here's the antibodies. And who makes the antibodies again? B cells produce plasma cells. Plasma cells produce antibodies. And so here we have an antibody attached to our germ, our bacteria. And then we have the C1 type of complement, C1. It is a complement, it is a protein. What will it do? As you can see, it is attaching to these two, and it's saying the following. It says, acts as an enzyme, catalyzing a series of reactions involving other complements. So other complements can come around. And then we move to this, where we now have yet another type of complement attached. Okay. So what we're doing is basically making that cell wall of the bacteria more and more visible. If we had an invisible plane and we wanted to make that plane more visible, we could do what? Get stuff attached to it that glows. And therefore, if you can't see the plane, but you can see the glowing light that is attached to the plane, then you might as well be able to see the plane as well. Okay? That's exactly what this is like. So we can't see the germ so well as an immune system, so we're going to attach chemicals to the plane to make them more visible. Okay? That's how it works. So that's the classical way. Right, so C1, complement, attracting C2, C3, C4, and ended up making C3b, which makes the cell wall more visible, more palatable. Okay, next slide. Okay. Now we're looking at the alternative way. In the alternative way, it's a slower method to expose the antigen. So a slower method. Okay, so here's the slide for that. Again, do keep these factors in mind. So now we're going to look at page 783 again, but this time the slow method. Okay, here's how that works. The alternative pathway is important in the defense against bacteria, some parasites, and the virus-infected cell. In this case, we're doing what? The alternative pathway begins when several complement proteins, notably this properdin, so properdin, interacts with the plasma. This interaction can be triggered by exposure to foreign material such as the capsule of the bacteria. So this chemical, along with the exposure to chemicals coming off the cell, because the bacteria does have a cell wall and it has products, the end result is again what? The attachment of the C3b to the bacteria's cell wall. So regardless of how we do it, either we have an antibody attaching to the cell wall or we recognize stuff coming off the cell wall. So let me say that again. This interaction can be triggered by exposure to foreign materials such as the capsule of the bacteria. So we shed skin. Well, if we can identify stuff that we are shedding, we can still react to that. So it doesn't have to be directly the skin, but stuff that we are releasing. So let's go back to the invisible plane example. Instead of detecting the surface of the plane that's invisible, we can actually do what? Find the exhaust from the plane. So if we can find the exhaust trail of a plane, even if we can't see the plane, but we can see the exhaust trail of a plane, we can do what? Attach something to the exhaust trail of the plane and pretty much follow the plane from the exhaust. Okay? It's very similar to that. Next slide. Now it says, both pathways end up doing what? So regardless of whether we use the classical pathway where we stick antibodies on the surface, or we actually use the alternative method and find the surface in a different way by the exhaust trails, if you want to use that. Either way, we activate C3b on the surface. Now, what will that do for us? So now we're going to go further on page 783's picture. Here's what it'll do. Once we have that, we're going to have a process called opsonization. Opsonization. Okay. So we're going to attach and create a process called opsonization. Now, opsonization is the enhancement of phagocytosis because we're now making the surface of that cell much more visible. Let me read you the box from page 783 on opsonization. It is the process of coating that germ with complement. So let me read you this. A coating of complement and antibodies both attract phagocytosis and makes the target easier to engulf. This enhancement of phagocytosis, a process called opsonization, occurs because macrophages contain receptors that detect and bind to complement. So if I'm able to get more complement onto a germ and my macrophages are able to detect that complement, then I'm able to engulf and get the germ. Okay, so to summarize that point, opsonization is the ability for the macrophage to enhance the phagocytosis because it recognizes the complement better. So if I can have more complement and more macrophages recognizing that complement, I'll have a better opsonization. The last bullet on page 783's picture is histamine release. Now, histamine is something that actually increases the blood flow to that inflamed area. So if you have an injury area and you have inflammation, the histamine actually increases blood flow. What is the advantage in having increased blood flow? It means more white blood cells will get there. So you will have more macrophage action if you have more histamine release. Let me show you that on the next slide. So by having a more inflamed response, I can bring more of my macrophages there to kill off cells. That's what that slide actually says. Next slide. So these are the classic issues with inflammation, and you've learned this in 2401. So in 2401, you came across swelling or edema, redness, heat, and pain. And all of this basically brings more macrophages to that injury area. Next slide. Okay. Don't care too much about this. We basically have said that. So what does that do? Brings more immune system there. That's what this line right here is basically saying. Mobilization of systemic defenses, local defenses, basically bringing your white blood cells there. Keeps the germs from spreading elsewhere, and it repairs that area. Again, just the general picture. I don't care too much about this one. Okay, so we've already talked about that. We've already talked about that. Here's a picture version of the same thing. So here's a little pin or an injury releasing mast cells releasing histamine. And histamine is what will actually cause inflammation and swelling and increased blood flow to that region. So it increases swelling and blood flow to that region. The slide continues with this picture. Dilation of blood vessels, which means what? Increased blood flow, increased vessel permeability, clot forms, and now here's where I said that what does it do? It brings in more of your white blood cells. And it says it in picture form. I said it in words. So it increases bringing in more white blood cells. White blood cells will release more cytokines. Cytokines will make, and notice what word I see here, macrophage, which basically means it stimulates level 2. Now, when I stimulate level 2, what does it say over here? Activation of specific defenses. So what is that level? Whenever the author says specific defenses, we're talking about level 3 and 4. So, in essence, complement is good at doing what? Complement is good at stimulating your level 2, level 3, level 4. Because by stimulating level 2, you're helping level 2 present to level 3. You're helping level 2 present to level 4. So levels 2, 3, and 4 improve. That is what this slide actually says in a nutshell. Okay, next slide. I really don't care too much about this. General feeling, general definition only. Same is true for fever. Oops, let me back up for a second. So general information right here. Fever, same thing. Now notice, I see again what? Interleukin. Interleukins are what? Cytokines. So when you do have a fever, you are again stimulating who? Macrophages. Now what does this slide say according to Nemesh's levels of 1, 2, 3, and 4? Interleukin belongs to who? Level 1. Macrophages belong to what? Level 2. This slide is saying that a fever, by way of interleukins, will stimulate level 2. So if level 1 stimulates level 2 can stimulate level 3 or 4. Okay, that's how it works. Next slide. Okay. We've already talked about this. I've mentioned words like specific, immunity. I should add what word here? Memory. So specific, immunity, memory. Remember it's specific because it responds to specific antigens. And it's what levels? 3 for B cells, level 4 for T cells. Okay. So yes, it is part of Nemesh's 4-level chart. Next slide. We've already talked about all this. T cells are what? Cell-mediated immunity. They are for what? Inside the cell. So I've already mentioned this. Inside the cell. B cells are antibody-mediated. And they are for outside the cell, like bacteria. Next slide. Now, we have 4 kinds of immunity. The innate, present at birth, adaptive, after birth, active, and passive. Now, innate is okay. It means present at birth. Adaptive, after birth. That's fine too. The 2 that I'm most concerned with are active and passive. Active means that you yourself have gotten sick and made what? Antibodies against the antigen. So active means that you were exposed to the antigen, you got sick, and you recovered by producing your own antibodies. Passive, on the other hand, is that you got antibodies from somebody else. So you got it from someone else. So when you get vaccinations done, and vaccinations have a weak form of the germ or the dead form of the germ, and you make your own antibodies, that would be active. If you get your antibodies from mommy through breast milk, then that would be passive. Okay, we're going to go look at each one of them. Now, on a test, you are going to be using this 4-term language, such as naturally acquired active immunity. Okay, so that's a 4-term language. Naturally acquired active immunity, artificially induced active immunity. Make sure you're aware of those 4 terms. Now let's look at the first one here, naturally acquired. Naturally acquired means you got it through the exposure. So you got sick and got it, or you got it from mommy's breast milk. That's natural. Artificial is through a vaccination. So when you get vaccinated for a flu shot, that's artificial. Okay, next slide. We have passive. Passive can be naturally acquired passive immunity. So naturally acquired passive is what I said earlier, antibodies from the mom through breast milk. That is natural. So naturally passive. Why is it passive? It's passive because you did not make the antibodies. Mommy made the antibodies and gave it to you. So anytime you see passive, it means somebody else made the antibodies but gave it to you. Now, the first 3 that we've done, pretty much everyone has come across. You've gotten sick and you made your own antibodies. You got antibodies from mommy, or you got antibodies from making it by way of getting sick through a vaccination. The one that is unique, and I know very few people on the planet that have had that done, and I have had what's called artificially induced passive immunity. That means I actually got injected by somebody else's ready-made antibodies. Now that's not the same thing as a vaccination. A vaccination is when you get a germ either dead or weakened, and then you make your own antibodies. That's a vaccination. In this case, I got injected with somebody else's antibodies. And that's how I got protected in my visits overseas. So somebody else's purified antibodies were given to me. That is called artificially induced passive immunity because it's somebody else's ready-made antibodies that you're injecting. There is a risk, but I had to take that risk. Okay, now, any questions about those 4? Be aware of the 4-term language. So 4 words, 4 terms. Now on page 785, the author has put a chart for you. Basically the same thing that we just went over. So this chart is a pretty good one. I would definitely keep this one in mind. And the examples that go with it would be nice to have as well. So what is active? What is passive? This one is common. That one's commonly understood. So is this one by mother's breast milk. This is the only one that is unique. And very few on the planet have had that done. And I have. Okay, draw a line. We now go to the next slide. Okay, now this slide and the information that goes to towards the end of the chapter is unique. Now, we already know much of this, so I'm going to skim through it. We already know that T cells and B cells respond only to a specific antigen, and they ignore all others because B cells, level 3, T cells, level 4, are specific. Okay. Then we have versatility. Because these lymphocytes, B cells and T cells, are very broad, because remember, we're making so many different variations of them. B cells are very unique. They make specific plasma cells. Specific plasma cells make specific antibodies. Even the T cells, as we saw, came in various forms. So each fights a different type of antigen. Active lymphocytes clone itself to fight that specific antigen. So you have many, many versions of the same kind of lymphocyte in you, and we call them clones. So clone A is only for antigen A. Clone B is only for antigen B, and so on. So you have a lot of versatility there. So many, many types of lymphocytes for many, many different types of antigens. Okay, so we can make zillions of different possibilities because of that. Next slide. Memory. We already know that because we're talking about level 3 and level 4, we know we're talking about memory. And memory goes with specific, it goes with immunity, which means the next time I get the exact same germ, I'm ready to fight. And I usually recover better the second time around because I already had memory cells for it. The other term that you see there is tolerance. The immune system ignores normal antigens. Now this is where many of your other diseases come around. So if you understand what I'm getting at, I'm talking about autoimmune diseases. When you start losing what's called the self-tolerance, that's where your autoimmune diseases come into play. Okay. So immune system ignores the normal antigens, it ignores your self-antigens, hence your self-tolerance. Next slide. We've already seen this. You've seen this already twice. So T cells go with cell-mediated immunity, B cells go with antibody-mediated. So we already got that. Next slide. Okay, this is a picture version of what we talked about. Now, this one is a very nice one to show you how everything works. This is your T cell row because it says cell-mediated immunity. Now, do you see this right here? It says phagocytes. That's actually belonging to what according to Nemesh's picture? Level 2. Now, are you telling me that level 2 can actually talk to level 4? Did we not already say that earlier? You bet. So level 2 can actually talk to level 4. Likewise, level 2 can also talk to level 3. So 2 to 4, 2 to 3. It also turns out that, as you can see from this picture here, antibody-mediated is B cell. This is T cell. So can 3 and 4 also talk to each other? Yes. So on your chart, what type of relationships are interactive? 2 to 3, 2 to 4, and 3 to 4. That's why that chart's so important. So again, 2 to 3, 2 to 4, and 3 to 4. And in the end, whether I am doing it by way of antibodies or by way of the killer cells, and I'm talking about the cytotoxic cells, I will destroy the germ and probably the antigens along with it. Okay. Next slide. Now, you've already seen all of these. So this is a rehash of the slides that we've already talked about. So I'll move forward. Okay, so we've already talked about those. Next slide. We talked about this concept as well, antigen presentation. Now, there's one catch here, and that is this bottom portion. The bottom portion is talking about MHC proteins. Every cell of our body has cell membrane markers. You learned that in 2401. We call them membrane proteins. And they played roles like receptor, channel, adhering or anchoring, and one of them was cell ID. And that is the one I'm now using here. So MHC, major histocompatibility complex, is related to the membrane glycoproteins. Now, why is that a big deal? Because that is how we identify each of us. So when you watch shows like CSI and they're doing a genetic makeup or they're trying to identify a cell, they're doing it through this method. They're matching the different types of surface antigens. And the more of a match, the closer you are. So your parents are closer to you. They're not exactly you, but they're closer to you. An identical twin would even be even more closer to you. Okay? So notice it says it differs among individuals, and an identical twin would be the closest possible match. Not exact, but closest possible match. And again, it's genetically determined. That makes sense. So it's genetically determined. Now, why is that such a big deal? Because that is what your immune system, when I say immune, I'm talking about T cells, B cells. That's what they're really responding to. Next slide. This slide says that there are two classes of MHC proteins. Class 1, found in membranes of all nucleated cells. That is how the body knows this is Nemesh's cells, this is a germ. Okay? So found in all membranes of all nucleated cells. Then we have class 2, found in membranes of antigen-presenting cells. Now, this line right here is an important one. On a test, you will need to know which cells are antigen-presenting cells. I'll give you the two major ones. One is macrophage. Because remember, I already told you that level 2 can talk to who? Level 3 and level 4. So antigen-presenting cells are the ones that present the germ. And in this case, macrophages are one of your antigen-presenting cells. Okay? That's very important. We also saw a moment ago on a picture slide that level 3 can talk to level 4. So B cells can communicate with T cells. So B cells are also antigen-presenting cells. So if someone asks you on a test, what are the two major antigen-presenting cells? They are macrophages and B cells. So macrophages and B cells are your two main antigen-presenting cells. You could look at it from a point of view of what are they talking about, or who are they sending it to. Macrophages, after they chew up the germ, they send it to level 3 or level 4. So macrophages are antigen-presenting cells. Level 3 can talk to level 4, so that's also an antigen-presenting cell. Okay, let's go back to class 1 again. Normally it says T cells ignore normal peptides. So T cells will ignore normal ones. So if you have normal markers on your cell surface, your T cells will ignore that. However, if it finds an abnormal one or a virus one, it's going to destroy that cell. And remember, T cells are intracellular. So if they find any remains of stuff that's bad on the surface of your infected cell or on the inside of your infected cell, it's going to destroy it. Now, that takes me to page 789's picture. So page 789. We're going to go through this picture. It is important. Now, notice I am talking about an infected cell. So this is an infected cell. What do we have here? We have antigen presentation by a class 1 MHC proteins is triggered by viral or bacterial cells. So here's a virus. It has gone in and taken over this cell. Okay. The infection results in the appearance of abnormal peptides in the cytoplasm. So now this infected cell has some bad stuff in the cytoplasm. It's a germ stuff. Well, what's now going to be presented on the surface? Not only is the germ inside the cell, the infected cell, so not only is it inside, but it's also presenting a dirty part of the germ on the surface. That is what your B cells and your macrophages will go after. Okay. So if you got some dirty stuff on the surface of your jacket, okay, it will detect that. Okay. It will detect that. Okay. Next. Okay. Here is the Class 2 story. Class 2. Now, instead of going over this slide, I want to show you a picture that goes with it. So here's the picture that goes with this slide. Here is now your antigen presenting cell. This can be either a macrophage or a B cell. So either a macrophage or a B cell. Let's say that this is a macrophage. What will it do? It'll find your germ. So this macrophage will find the germ. It will chew it up with digestive enzymes. And then it's going to present that germ piece to either level 3 or to level 4 if it's a macrophage. Or if it's a B cell that we're using here, it will present it to level 4. So 2 can talk to 3 and 4, and 3 can talk to 4. Okay, next slide. Okay. I really don't care too much about this slide. Okay. I've always been using this one as an example, so I really don't care too much about the slide. Macrophages are antigen presenting cells, so we've already talked about that. Don't care about that. And now we have antigen recognition. Okay. So binding occurs when MHC protein matches the antigen. So now what we're now going to see is, how do I recognize the germ once it is presented to me? So how do I recognize the germ once it's presented to me? Okay. Now, we have what's called CD markers, and they're found on T cells. So we have CD markers marked on T cells. Now the slide I was on a moment ago, I don't care too much for that, but I do care for this slide I'm on now. Okay. So this slide is important. We have CD8 markers. Those are found on your cytotoxic cells. So your cytotoxic T cells are CD8 markers. Your helper cells are CD4 markers. And matter of fact, you're often able to identify the type of T cell solely from the markers. Now these markers are going to grab hold of your antigens that you're presenting. So these markers will grab hold of the antigens you're presenting. Now it's good to see this list here. CD8s respond to the class 1 markers. CD4s are responding to the class 2. So be aware of this slide. That's why I said this slide is important. So CD4 goes with class 2, CD8 goes with class 1. CD4s are often called helper T cells, CD8s are called cytotoxic. So this slide is important to keep in mind. Next slide. I don't care too much about that, so we'll move on from there. I don't care too much about this slide because it doesn't help me. I'd rather show you a picture. So we're going to see a picture that's coming up. Same as for this slide, it really doesn't help me. I want to see the picture that goes with it. So this is a very important picture. I am now on page 790. So it's page 790, and this is a very important picture for page 790. Let's study this picture together. Here I have a virus or a bacteria, whatever it is, virus or bacteria. And notice I have an inactive form of CD8, inactive. Once I am presented that germ, it will then do what? activate it. So you don't have active floating. They're actually inactive at first. It is the recognition or presentation that's going to make the difference. So the recognition or the presentation. Now, who could this guy be? This guy right here can either be a B cell or a macrophage. Okay. That's why you need to be aware of that. So this could be either a macrophage or a B cell. Then, what do I have? I've got the presentation here. It activates it, and now I have two forms. I have the active cytotoxic, that's what that little C stands for. Here's the name on the top. Cytotoxic T cells. So cytotoxic T cells. I have the active version and a memory. Why memory? Because next time I come down with that same exact germ, I am ready to go. I don't have to be delayed. And what will this cytotoxic T cell do? It's going to kill that germ, whatever that germ is. It's going to kill it. Now, this is the story on co-stimulation. So the story on co-stimulation. So before activation can occur, a cell must be physically or chemically stimulated by way of this antigen and this marker. So almost like turning it on. That's what this co-stimulation is, turning it on by presenting that antigen. And that should be in between here. Okay, next slide. All right, and we already know that the active form of the cytotoxic T cell will do what? Kill the cell. And you have words like apoptosis that you learned, disruption of the cell's metabolism, and breaking the cell's membrane. Okay? And there you go. So it's destroying the cell's membrane, killing the cell, lysing the cell. Okay, that's number 4. We've already talked about this. Okay, so these are ready to go. These memory cells are ready. They stay in circulation, and they're ready to go for the next time around. And they will last a long time. Suppressor T cell, not much to say about that. Helper T cell. This is a very important one. Notice, helper T cells remain in reserve. And I said earlier that helper T cells are the ones that are going to connect levels 2, 3, and 4 together. And we'll see that. So helper T cells are a very good thing to have. They keep your immune system strong. Next slide. This is now showing you what happens with the helper T cell. Same type of story. This time around, we're dealing with the class 2, though. So this time we're dealing with class 2. Again, I have an antigen-presenting cell. This time it can be a macrophage or it could be a B cell. Again, it doesn't matter what we're talking about. So 2 can talk to 3, 2 can talk to 4, 3 can talk to 4. And same type of story again. And all right, again, as usual, we have what available? We have memory helper cells ready for next time. Now this time around, look what it can do. It can produce cytokines to tell other cells what to do. And that's why helper T cells, or that's what they call them, helper T cells, why they're so good. They can talk to others. They can tell both. Now let's read this line carefully together. Cytokines that stimulate both cell-mediated and antibody-mediated immunity. Now, why is that paragraph so important on this slide right here? Because it says that these cytokines will stimulate levels 3 and 4. So it's going to stimulate levels 3 and 4. Now, what I just said is already on this slide. Look what it says here. Attracts and stimulates more macrophages. So what level is macrophages? Level 2. Skipping to number 4 here. Promotes activation of B cell. What level is that? 3. Number 3 here. Attract and stimulate more cytotoxic T cells. What level is that? 4. So do you now see that helper T cells stimulate levels 2, 3, and 4. That is why this slide is a good summary. It stimulates levels 2, 3, and 4. Okay. Now if you turn the page to page 792, the author says it in picture form. So here's a picture form of what we have been talking about. And then the next picture talks about how it stimulates and helps others. So this is picture B. Okay, next slide. So again, don't forget, helper T cells work with level 2 macrophages, level 3 B cells, level 4 T cells. Next slide. Okay, now we go into the details of B cells. Again, most of this is redundant. We've already talked about B cells being antibody mediated. We already know that it makes specific antibodies. Now what it does do is make millions of various versions of it. Various versions of it, millions of various versions. And each version goes with what? A specific germ. Now, this slide is important to keep in mind. So this slide is important to keep in mind. Okay. Now, since I don't like to have you write a lot of slides, I'd rather have you study the pictures that go with them. So this slide is important, but the picture is better. And here's the picture version of it on page 793. So again, here too, I start with what for a B cell? An inactive form. And what comes in? The antigen. It stimulates this B cell. So an antigen stimulates the B cell. The B cell then becomes active. Now the B cell doesn't do anything directly. What the B cell does is it makes plasma cells. So we're going to skip to picture 2 now and show you that what are we doing here? We're activating the B cell. In this case, who did it? A helper T cell. Remember I told you that level 3 can talk to level 4? Well, here's a co-stimulation. And again, I'm using what word here? Cytokine. Now to clarify, we have the 2 cytokines that we spoke of. One was interleukin and one was interferon. We've seen both. I'm now going to clarify the definition. Interferon comes from the virus-infected cell. Interleukin is actually a communication between level 3 and level 4. Now what does an interleukin do? It tells, like a police officer would, to the police station and say, send out an all-point bulletin. And that's exactly what this is like. So this is a all-point bulletin, in this example, coming from a helper T cell telling a B cell, hey, I found this germ, you need to be aware of it. So it's sort of like a memo coming from within the police department, sort of like the wanted posters that you see at police stations. You got the FBI most wanted list, the state's wanted list, that kind of thing. And everyone always looks at each of these lists to see whether they got a bad guy in their neighborhood. Well, that's exactly what this is like. It's an all-point bulletin called interleukins, coming from level 3 or level 4. And in this example, it's coming from level 4. So level 4 is telling level 3, I found this germ and what you should do with it. So now let's go to bullet number 3, page 793. Here we see once we have stimulated and activated the B cells will produce plasma cells. And the plasma cells will then make antibodies. Now there's one big mistake in this picture. And they're showing you these antibodies all looking the same in terms of a Y. Okay? Now, a given plasma cell will produce the same margins up here, these little antigen antibody binding sites. But different plasma cells will produce different types of antibodies having different antigen antibody binding sites. We'll see a better picture of that in a moment. Okay. Now, let's go to this line. Plasma cells produce antibodies. We know that. We also have memory B cells. So just like memory T cells, we have memory B cells, and they're ready for the next infection. So there's that early advantage there. Next slide. This is where I was talking about. Now, I don't like the picture here, because there isn't one. I don't like this. We're going to go look at a picture that goes with it. So I'm going to skip down to the picture and then come back to where I am right now. So here's the picture that goes with this slide. Now, here's your typical antibody. You have the heavy chain and you have the light chain. So heavy chain, light chain. You have a variable region. So you have your binding site and your variable region. Your binding site and your variable region. And this part is going to be the same. And that's why it's called constant segments. So you could say that this part right here is all the same each and every time. And what's different is this variable region. And so each plasma cell will produce an antibody uniquely having a specific binding site. So no two antibodies are the same unless they're from the same plasma cells that created it. So if I'm plasma A, then I'm making antibodies with antigen A markers at the top. If I'm plasma B, I'm producing little antigen binding sites for things for antigen B. So A goes with A, B goes with B. Now let's go back to where we were. So be aware of this. This is the part that goes to the macrophage. This part sticks to the macrophage. This part sticks to the germ. So one more time. Germ sticks here. Macrophage sticks down there. Okay, returning to where I was, this is the slide I was on. Moving forward, I have the 5 types. So the 5 types. IgG, IgE, IgD, IgM, and IgA. The most common form is IgG. That you do need to know. So IgG is the most common form. The oddball that looks different is IgM. And IgM is considered a pentamer because it looks like a pentagram. And you'll see that in a moment. Or like the pentagon. Okay, so IgM is a pentamer, okay, like the pentagon. Now each one of these has a different function, and we'll see that in a moment. Next slide. We've already talked about this slide. We said that every single one has an antigen binding site, and it's specific, right? So each antibody has a specific antigen binding site. Okay. Next slide. So each antibody has a specific binding site. Okay. This slide, not too much concerned with that either. Okay. So not too concerned with that. Next slide. This one is important. Okay. So this one is important. Now, each antigen has a group of markers called haptens. Okay. So if you looked at a series of antigen markers on a germ, or on any cell for that matter, each antigen had, markers, each of these markers are an array, a group. So if I had circle, 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 that would still be an antigen. But it's what? Circle, circle, circle. If I had a circle, circle, circle and an X, Y, Y, then that would be a different kind of antigen because the markers are slightly different. Now look what it says here. antibodies produced will attack both hapten and carrier molecules. If carrier is normal, antibodies will attack, so it says antibodies attack normal cells. For example, someone with a penicillin allergy. Okay? So if you take penicillin and you're allergic to that, well, guess what? Your immune system will detect not only the penicillin coming into your body, but also it will start eating away your normal cells as well. Because for some reason, your body considers penicillin to be sort of like your normal cells. Right. So that's the key thing to keep in mind with this term right here. Now, the term hapten and epitopes are on page 794. So that's an important page to look at. So page 794. They talk about haptens, epitopes. So haptens and epitopes. So you could say that a hapten is a portion of an antigen. So I might have circles and boxes and X's, but if they're not in the same order, it's not the same antigen. Okay? So there are variations of an antigen, and that's your hapten. And some people have variations in their haptens or their antigens, and they mimic something else. So they'll mimic something. Okay, this picture I've already shown. This is another version of that. And here's what it looks like. So here is a germ and the antibody. And as you can see, the antibodies are sticking to specific antigens. So the antibody is sticking to specific antigens. So antibodies bind to portions of an antigen called antigenic determinate sites or epitopes. So where that antigen is located, that location, that part is called an epitope. Okay, next slide. Now here's the difference between an antigen and a hapten to give you an idea. Okay. So here's a complete antigen, and see how this part right here sort of mimics this thing. So a portion of an antigen is a hapten. That's a nice definition. So a portion of an antigen is a hapten. You could have an antibody responding to both portions of the antigen, and in this case, look, is attaching to a carrier molecule, but it's also attacking the hapten. And that's where you have problems, because if you have a carrier molecule looking very, very similar to an antigen because it has a similar piece, then your body is going to respond to it. Okay, next slide. We've already talked about this. I just want to make sure you understand that antibodies are also called immunoglobulins. They're found where? Remember, chapter 19? They're found in the plasma portion of our blood. Okay, so they're found in the plasma portion of our blood. All right, next slide. Okay, now, we have 5 classes. Each one of these slides that are now coming up are talking about the different versions of antibodies. Now, I do want you to look at these 5. So all 5 slides, make sure you look at them. So that's 1. This is 2. This is 3. This is 4. And this is 5. So make sure you do go over those 5 classes. Now, as a summary, the author has done a chart for you. So we have IgG. I told you it was the largest and the most common. Then we have IgE. Now, IgE is often the allergic reaction one because it goes with what's called a mast cell and histamine. You can see that right here in the section. It says basophil, mast cells, and you already know that basophils go with allergic reactions. So basophils, mast cells, allergic reaction. Then we have IgD. Next slide. IgM, this is the pentagon or pentamer I mentioned. And then we have IgA here. Okay. Be aware of all 5 classes. Okay. Next slide. These are the steps basically that go with antigen-antibody complex. If you look at them carefully, you will find that we've already talked about that. It's very similar to how that phagocytosis comment worked. The only new term that we have to keep in mind is agglutination. And again, that is very similar to the blood clotting. Next slide. Now comes a new thing about immunity in first response, second response. I do not care for this language that you see on the slide, but rather I want to discuss the pictures on page 796. So we're passing this slide up as well. So that slide, this slide is now going to be shown in picture form. And that's this one. So we have IgM. So you've got infected. There's your day 1. And now we have our weeks. So we have IgM followed by IgG rising. And that's your first response, first attack by that germ. Then, when the second response comes, look at the second time around. So this is the second time around that exact same germ came. IgM still takes its sweet time getting up there, but IgG is way up there because of the readiness response. So that readiness response. And they are mentioned in these slides. So it says IgM is less effective and is produced faster than IgG. It is produced faster in what? The first response. So in the first response, in the first response, IgM kicks in, but it's less effective. So in the first response, IgM kicks in, but it's less responsive. Okay, so it kicks in, but less responsive. Then in the second one, you'll see that the IgM is much faster in responding. Okay. So when you have a memory issue, IgG rises very quickly and remains elevated, whereas IgM production is quicker, but it's slightly extended. It takes a longer time to make that happen. So those 2 slides I showed you are important. Next slide. We've already looked at this, but I want to show it to you in picture form now. So here's our response. Day 1, look what happens. Neutrophil kicks in first. And we've already learned that, right? So neutrophil is your first sign of an infection. And then after neutrophils, who's next up there in line in terms of quick timing? Natural killer cells. Then who comes in shortly after in this purple line? Cytotoxic cells. Along with who? The macrophages. Why? Because macrophages talk to who? Talk to the level 4, right? So macrophages are talking to level 4, cytotoxic. And of course, right about here, you see what happening. As T cells kick in, you see B cells kicking in as well. And when B cells finally kick in, you also see a rise in antibody. Okay. So this picture is definitely worth keeping in mind. Neutrophils are first, natural killer cells next, then you see macrophages kicking in. And notice, 1, 2, 3, they're all part of what? The first 2 are part of level 1. That one, macrophage, is level 2. Then, as you can see, who kicked in? Right here. After the green, we see purple. And purple is level 4. Level 4, once you see level 4 kicking in, who do you see level 3 kicking in? And level 3 is going to keep your antibody levels high and protect you for a long time. So that picture is worth keeping in mind. Okay. Don't care for that? Okay. Here's your virus. Nothing more I can say, same thing. Now, who is the antigen-presenting cell here? Macrophages and B cells. So macrophages and B cells. Next picture, same thing. Okay. Who do we have here? Now, these pictures are a summary of what we've already talked about. So it is worthwhile looking at. So page 798, 799 is worth looking at. Now, when you do look at these three pages or three pictures, page 798, 799, you're going to find a common theme. Matter of fact, let me back up a slide and you'll see. If you look at this row right here, activation of helper T cell, activation of B cell, antibody, and I go over to the next slide, you're going to see the exact same thing. So you're not memorizing different pictures really, you're just memorizing the commonality among them. Okay, next slide. This is a summary at the bottom, worth looking at. And this is a summary at the bottom, worth looking at. Okay. And I did mention these to you. Dendritic and Langerhans, Kupffer cells, fixed macrophages, they've been mentioned. Okay. Next slide. Now, this slide takes off the final aspects of the chapter. We have fetus can produce immune response after exposure to antigen at about 3 to 4 months. So the fetus can produce an immune response. Okay. All right, so there is a fetal response going on as well. Okay. Now this slide's important. Maternal IgG antibodies through the placenta. So yes, you are getting your immune system from mommy through the placenta. Okay? So just like breast milk mentioned here at the bottom. So notice they are both passive. Okay? So they're both passive. Okay, next. All right, this slide is worth keeping in mind. Infant produces IgG antibodies through exposure to antigens. Antibody, B cell, T cells levels slowly rise to adult levels at about age 12. And that's where you get your normal protection, by age 12. Okay, so normal resistance by age 12. That is why kids very, very young and the very, very old have issues with their immune system. We've already talked about this, so this is worthwhile keeping in mind. Okay. So we talked about interferons, interleukins. Okay. So this slide is important. I've already talked about it. So interleukin is a communication between B cells and T cells. And I said it by saying it's a communication between level 3 and level 4. Here's more information about that same thing. Okay, so that was interleukins. Okay. Now when it comes to the test, I would keep this slide in mind and the next one in mind. So keep those two in mind. We've already talked about the alpha interferon, beta interferon. It was mentioned earlier in the picture. Okay. Tumor necrosis factor. This slide is one to keep in mind, just a basic definition. I don't care for this one, and I don't care for that one. This one is a new topic. It's autoimmune disorders. And remember I said that we are having more and more diseases listed as autoimmune. So we have autoimmune disorders, immunodeficiency diseases, and allergies. Next slide. Autoimmune. Notice, diabetes is listed as what? An autoimmune disease. That is something I do want you to know. So autoimmune disorder and diabetes, the insulin-dependent diabetes mellitus, is an example of autoimmune. That's something to keep in mind. Okay, then the next slide, we talk about SCID, and I mentioned this with David the Bubble Boy. Severe combined immune deficiency. I also mentioned AIDS in my story. So AIDS, HIV. They are immunodeficiencies because they are actually destroying various levels, such as levels 3 and 4. If you have HIV and have AIDS, you're going to have a problem with your helper cell. And if you have SCIDs, you've lost level 3 and level 4. Okay. So that's something to keep in mind for this slide. Next slide, allergies. We all have them. And what are we responding to? We're responding to those weird antigens. So some of us have responses to certain antigens. Some people have allergies, some people don't. Some people develop them later in life. These are now 4 categories of allergic reactions. So type 1, type 2, type 3, type 4. Make sure you have a 1-sentence example of each. So 1-sentence example for each. And you will find them on page 804. So starting with page 804, you're going to find them. So make sure you have a 1-sentence example for each. Okay, so 1 example for each of those. So that was type 1. It's anaphylactic shock. That's something to keep in mind. Okay, so anaphylactic shock. Okay, so we're dealing with that type A. Then we have antihistamines. Okay, so let me make sure we're all together. So what I'll be expecting you to do with this is have a 1-sentence example of each. And then the next thing I talked about was the anaphylactic shock. And that goes with picture 1, picture 2. And the problem with that, with having an anaphylactic shock, is this. Your capillaries will dilate. You'll have increased capillary permeability. That means what to us in chapter 21? You're going to have a sudden drop in blood pressure. Okay? So capillary dilation and increased capillary permeability is going to cause a drop in blood pressure. And that's not good. Also causing airway constriction, so you have trouble breathing. Mucus secretion, again, having trouble breathing and pain and itching. So that is why you take things like antihistamines. So that is why you take antihistamines, such as Benadryl. So when my daughter had a big bump on her face from a mosquito bite, okay, or some insect bite, I had to give her Benadryl to cut down on that big swelling on her head, for that big bump on her head. Okay, so that's what I'm getting at. When you have allergic reactions, you produce histamines. Histamines can do all nasty things to you if you're very sensitive to it. And then you've got to make sure. Now that's why some patients with allergic bee stings, for example, they carry an EpiPen containing epinephrine so that your blood pressure does not drop. Because as I mentioned a moment ago, blood volume will rush into the capillary space, the cell space, and now all of a sudden you'll have a drop in blood pressure. You'll also have vasoconstriction of the airways, so that's why you want to take something like an EpiPen. Okay, next, we talked about antihistamines here. They'll actually block the mast cells from producing histamine and relieve some degree of sensitivity. Okay, stress and immune. Now, this was already discussed in chapter 18. So we saw this in chapter 18 with glucocorticoids. So that was already there. Okay, so we saw this again. So Nemish, are you telling me that chapter 18 on hormones is related to the immune system? You bet. Okay. It is a very strong connection. Now, look what it says here. Depression of inflammatory responses, inhibition of interleukin secretion. So what do you think if you're in a high stress situation? What do you think is going to happen to your immune system? It's going to drop. Look what it says in the middle bullet. Reduction in abundance and activity of phagocytes. So if you have too much of a glucocorticoid response, yeah, you are fighting your inflammation response. You're having that side of your story okay because you're able to deal with the stress. But at the other end of that stress story is that you are suppressing your immune system. So let me say that again with the last two slides. Look what it says. Inhibits immune response. Lowers resistance to disease. So yes, even though glucocorticoids is a good thing to help you with your blood sugar and your stress for blood sugar and glycogen response, but it actually lowers your immune system. So that's what these two slides are talking about. Next slide. What happens when you age? Well, like I said, when you're very, very young and when you're very, very old. When you're old, what happens? Increased vulnerability to infections and cancers. These are the reasons why. T cells become less responsive. Now, T cells are level 4, right? So T cells tend to go bye-bye in their reactivity. What about the next line? Fewer T cells reduces responsiveness to B cells. So now all of a sudden, what have you done? You've shot away level 3 B cells and their nice communication with level 4, the T cells. And it's almost as if you're having SCID kind of symptoms later in life with your immune system. Okay? So it's really, really important to keep up level 3 and level 4. And here it says immune surveillance against tumor cells declines. So you're more likely to come down with cancers because even that's not working. Okay? Now the last slide. Nervous system and endocrine. That too is related. So that means if you're always having a high response to your nervous system, it's going to affect your endocrine system. And your endocrine system, as we've just seen, also will diminish your immune response. So if you screw up your nervous system as you get older, you screw up your endocrine system as you get older, you're going to mess up your immune system as you get older as well. Pretty interesting. Okay. Now that concludes chapter 22. Make sure you look at page 805 on HIV and AIDS. So page 805 on HIV and AIDS. Make sure you look at that clinical box.